Hello, everybody, and welcome to another episode of Meet the Researcher. Today, I'm excited to be joined by Dr. Ramaprasad Srinivasan. Dr. Srinivasan is an investigator in the Urologic Oncology branch of the National Cancer Institute at NIH. His work includes developing treatment strategies and clinical trials for patients with both hereditary and non-hereditary forms of kidney cancer. Dr. Srinivasan, thank you for joining us.
Thank you so much, Josh. It's a pleasure having all of you here today to listen to me. I'll be talking for the next few minutes about some of the work that I have been doing and the group I work with has been doing. Focus of our work really has been to try and find alternatives to surgery in patients with VHL. Some of you may have heard one of my colleagues, Dr. Mark Ball, earlier in the week. He laid out the ins and outs of surgical approaches, especially as they relate to kidney cancer associated with VHL. As you know, a lot of the work relating to VHL has been done at the National Institutes of Health. I'm showing you part of the campus. It's a large campus, over 300 acres. Some of you have been here. For those of you who haven't, multiple buildings with state-of-the-art research laboratory, and what you see pictured in the foreground is a state-of-the-art clinical center where we take care of our patients. So this is the campus and the building that I was referring to a few minutes ago. So it's here that and a lot of the early work that went into identifying the VHL gene, studying patients with VHL, and really devising surgical strategies to optimize care of these patients occurred. So as you know, VHL-associated tumors are largely taken care of by means of local control. So surgical or ablative procedures are largely used to control VHL disease at this time. And what do these approaches do? They minimize the risk of metastasis, which is particularly true of kidney cancer, pancreatic neuroendocrine tumors, and pheochromocytomas that you see in patients with VHL. Sometimes these procedures are undertaken to control local symptoms, as with CNS hemangioblastomas or retinal angiomas, as well as endolymphatic sac tumors. And sometimes you control systemic symptoms or systemic complications, as in the case of pheochromocytoma, by surgically removing these tumors. Some of these tumors, especially kidney cancers, pancreatic neuroendocrine tumors, and pheochromocytomas, can metastasize, and when metastatic, they can cause problems that you would normally see associated with disease that spread beyond the organ of origin. There are no specific studies that deal with VHL metastatic disease as a whole. However, management of patients who thankfully are few and far between now, with improvements in early detection and surgical management of these patients. But when we do see patients with metastatic disease, they manage typically using the standard of care options that are available for sporadic or more common non-hereditary forms of the cancers that you see associated with VHL. So if you had a patient with metastatic kidney cancer and VHL, you would treat them with the strategies that are being used currently for treating patients with sporadic forms of clear cell RCC. So why should we even think of exploring alternatives? I mean, surgery seems to be doing fine for most people. Most people are well controlled with surgical strategies. So why are we looking for alternatives? I don't need to tell most of you why. As you know, many patients undergo multiple surgeries during their lifetime. And these surgeries aren't restricted to one organ. They encompass many, many organs. I've known people who've had 15, 20 surgeries in their lifetime, even more. With each surgical procedure, there is perioperative morbidity, complications from surgery. And as you do more and more, the surgeries become more challenging. There's gradual loss of function. If you keep operating on the kidney, you're going to lose kidney function or pancreatic and adrenal function. You're going to develop neurologic deficits. So many of these are far from desirable. And while we have effectively managed some of the aspects of VHL surgically, we're always looking for alternatives. We're always looking for ways to improve outcome in these patients. We are always looking for ways to improve the life of patients with VHL. As you all know, just because you've operated on a patient 10 times doesn't mean that they're not going to get new lesions. And so there's a lifelong risk of developing new lesions. So we should also be thinking of ways to minimize or reduce the incidence of new lesions if we can. So my group has really been focused on looking for systemic therapies as alternatives to surgeries over the last several years. I joined this group at the NCI 15 years or more ago. The group at that point was, largely focused on surgical approaches to VHL. There's a lot of basic science research, some of which I'll, briefly allude to, that I think has revolutionized the field of kidney cancer treatment. But many of those strategies and many of those strategies being developed in the lab, they're not necessarily being applied as well as we would like in the clinic. And that was really my role. That's when I came into the NIH and took this on as one of my primary missions. And when we started doing this, we tried to define what the goals of therapy are in patients with VHL. Some of the things that would be desirable, we thought, were delaying or avoiding surgery. So if we had a treatment option that would delay or avoid surgery, we thought that would be very, very helpful. Certainly, we know that early detection and surgical intervention, we prevented prevented metastatic spread of these cancers, and we would want any systemic therapy option that we applied to these patients to do that, to do the same. We would also hope that anything we have would improve quality of life, preserve organ function, and have very acceptable short and long-term side effects. And these are the goals. That doesn't mean that we're going to get there very easily, but I think if we can accomplish some of these goals, we've made some clear headway in managing patients and trying to lead them away from repeated surgical procedures. So how do we go about this? I think one of the basis for all this comes from our understanding of how VHL tumors really form, what causes them, and hence by understanding that, we can try and figure out how to intervene and potentially reverse the consequences of genetic and molecular changes that really lead to tumors in VHL. A lot of this work began in the 80s when Dr. Marston Linehan, Burton Zabar, and others collaborated to study VHL families at the NIH. A large group of families was studied, and by studying these families, they were able to identify that there was a gene on chromosome 3 that seemed to be very important in causing tumors associated with VHL. And what they really found was families who had VHL were affected in their germline, meaning every cell, they were born with it and every cell in their body was affected by changes, alterations in this gene on chromosome 3, which we now call the VHL gene. And we found subsequently that these alterations inactivate this gene. So the protein that's made from this gene is not functional anymore. And that we said was the root cause of tumors in patients with VHL. How do we go from a change in one gene to tumors? We don't have all the answers, but what we do know is that the VHL gene plays a very important role in sensing the oxygen needs of cells. Normally, if you have a lot of oxygen in and around a cell, then the cell shuts down a pathway called hypoxia-inducible factor pathway. And what the pathway really does is figure out how much oxygen there is around the cell. And if it feels like there isn't enough oxygen, it sends the nucleus a message, which allows it to generate new proteins that then compensate for this lack of oxygen. When VHL is altered, the cells constantly believe that there isn't enough oxygen around them. And so there is complete dysregulation of this process of oxygen sensing that would normally occur. And this dysregulation then leads to tumors. This is just one of the mechanisms by which tumors are caused. And abbreviation you see here, HIF, is for hypoxia-inducible factor. It's one of the key mediators of this process. Here we show you something called hypoxia-inducible factor 1 alpha. But we found subsequently through work done here and at Harvard and other places that a protein called hypoxia-inducible factor 2 is indeed a lot more important in causing kidney and other tumors associated with VHL. As many of you know, the Nobel Prize recently was awarded to Dr. Bill Kalin as well as his colleagues, Dr. Peter Ratcliffe and Greg Semenza, for work that really helped us understand oxygen sensing. Some of that work is very relevant to understanding how VHL tumors are formed and indeed how we can effectively generate therapies for patients with VHL tumors. This is just a cartoon that shows that a lot of the work that's gone into understanding the consequences of VHL loss have really resulted in the vast majority of what we call targeted therapies that have been used in the management of patients with non-hereditary or sporadic clear cell kidney cancer over the last year. So beginning in 2005 or so, a number of agents that target some of the downstream consequences. So you see here things like VEGF or vascular endothelial growth factor, MET, PDGFR. These are all things that go up when VHL is lost. While the process is mediated by activation of this protein called hypoxia-inducible factor 2 or HIF. And we have been able to target these downstream consequences of VHL loss over the years with a variety of agents which have been effective in sporadic kidney cancer. Initially, none of these agents had really been tried in VHL patients, but we and others did a variety of studies to try and understand whether those agents that are useful in sporadic kidney cancer would also be helpful in patients with VHL. After all, the biology is very similar, so we would suppose that they would be, but unless you do studies and show that there is indeed some benefit associated with these drugs, we don't know for sure how useful they are. Some of the studies I list here are studies done in the early days with inhibitors of angiogenesis, or inhibitors of vascular endothelial growth factor receptors that I briefly alluded to in the previous slide. A study in MD Anderson of sunitinib led by Dr. Yonash, another of pazopanib, a similar agent, again, led by Dr. Yonash. And we here did a study at the NCI of a drug called vandetanib. This is one of the largest studies of a drug in patients with VHL. And I'll show you briefly what we learned from those studies. But more recently, we've developed interest in this, in these molecules that we know can actually target a more vital pathway or an upstream consequence of VHL inactivation, meaning that we can directly target hypoxia-inducible factor with some of these drugs. And I'll go very briefly into these trials towards the end of my talk. But first, let's talk about what we learned from doing trials with angiogenesis inhibitors. I'm going to here highlight the results from two of these studies. These are two of the largest studies. The pazopanib study had 31 patients. The vandetanib study had 37 patients. And what we found from both studies that, yes, these agents are indeed associated with activity. So we could see some tumor shrinkage. That activity was best studied in patients who had kidney tumors. Both Dr. Yonash and I are kidney cancer doctors. And so that was quite naturally one of the major points of interest for both our groups. And we found that kidney cancers do indeed shrink in response to these treatments. We have much less information about pancreatic neuroendocrine tumors, pheochromocytomas, endolymphatic sac tumors. But none of these studies really showed much of an impact on CNS hemangioblastomas, which is disappointing. What we also learned from these studies was that just because these drugs are used with a fair amount of success and what we think is fairly in a well-tolerated manner to patients with sporadic metastatic clear cell kidney cancer doesn't mean that the same applies to patients with VHL. The VHL patient population is a very unique population. They have very different needs from patients who have metastatic clear cell kidney cancer. Many of the VHL patients, as you know, are very active, lead very active lives, are gainfully employed, and have another option. Patients with metastatic kidney cancer don't have surgical options. Patients with VHL do have surgical options. Despite the issues associated with surgery, there is that option. So we tend naturally to look at anything we're doing, compare it with surgery and see, is it worth going through all this for the potential benefit that you're getting with these systemic therapies. And what we found was a lot of patients in both studies found that side effects associated with these drugs quite difficult to tolerate. It was having an impact on their quality of life. Their day-to-day life was being impacted significantly, such that 57% in one study, 62 in the other, stopped their drug because of toxicity. And it's a very, very important lesson. I think we all learned from this that if we are to succeed and help patients with VHL, we need drugs not only that are effective, but also that have toxicity that can be well managed and that don't really impact too badly the quality of life of these patients. In fact, because of the side effects, there was a relatively short duration of treatment in both studies. In the Vendetinib studies, we allowed patients to continue on therapy for as long as it was helping them. And in Dr. Yanash's Pazopanib study, the plan was to stop after 6 months, but if somebody was benefiting, then they would continue. But what we found was on average, patients were only able to take this drug for 6 months or more. Many for less, although some patients took it for a lot more. We didn't know the long-term side effects because we couldn't treat patients for long terms. And we clearly didn't know what the long-term benefits were. We knew that the tumors would shrink briefly while they were on the drug, only to grow again when these drugs were stopped. We couldn't continue the drug for long periods of time because of side effects. So we didn't really know if these drugs were benefiting people in the long term. My gut feeling is they probably were not. And it was really unclear, and it was hard to study whether these treatments were preventing or slowing down the development of new lesions. So with this in mind, a lot of us are looking at new therapeutic strategies. I'm going to very briefly allude to one of these strategies that an investigator, a neurosurgeon at the NCI is very, very interested in. And this is using a drug called Propranolol, which is a very, very, very old drug. It's being used to treat a variety of conditions, most notably hypertension and symptoms associated with pheochromocytoma, tremors, arrhythmias, migraine, and so on and so forth. It's a very, very widely and easily available drug. What was noticed several years ago was that there's a condition called infantile hemangioma. And infantile hemangioma, as the name suggests, it's a abnormal blood vessel growth, such as what you see in patients with VHL, except that this is affecting infants. These infants don't have any VHL defect that we know of. And they can be quite disfiguring, as you can see in some of these pictures. They're considered benign tumors, but because of the extent and sometimes the location, they can be a challenge and clinically a problem. Patients with this children with these conditions were treated with this drug called Propranolol, and what they found was that it was very effective. You can see from these photographs that many of these children who received Propranolol actually had a very significant benefit with shrinkage of these tumors. There's some similarities between these tumors and the hemangioblastomas that people get in VHL. And because of this, there was some interest in trying to study this drug in VHL. And Dr. Chittiboyana, who's the neurosurgeon I was referring to at the NIH, his lab conducted some studies in kidney cancer that had no VHL or VHL defects, and found that these in the test tube or on a petri dish, Propranolol suppressed the growth of these tumors, and it did so, they think, by inhibiting this protein that I talked about as being very important, HIF2 alpha. And when they put these kidney tumors in mice and treated them with Propranolol, they found that, again, HIF2 alpha was being inhibited and that the tumors were shrinking. So based on this, they've designed a study to study Propranolol in a much more formal way in patients with CNS hemangioblastomas. But before they did the study, they also looked at patients who, for one reason or another, were on Propranolol, not necessarily to treat these tumors, but for one of the other indications that I mentioned earlier. And they had 66 tumors in 3 patients, of which 25 had evidence of growth. And they looked at those patients when they did, these patients, they looked at a period when they were on Propranolol and then got on Propranolol and found that the growth rate was much quicker when they were on when they were not on Propranolol, and that the Propranolol seemed to slow down the growth. This was a retrospective study. It's not very robust, but it certainly provided additional impetus for Dr. Chittiboyana to design this study. The study, I believe, is now open, but I'm not, I'll check and have to get back to you to make sure if it's indeed open. It'll allow participation of anybody who's over 18 years old, have demonstrated progression of 1 or more CNS hemangioblastomas. And obviously, this is for patients with VHL. And the scheme is laid out here. I'm happy to answer questions about this trial later if anybody has any questions. More interestingly, through a strong collaboration between industry and academia, more recently in the last 3 to 4 years, very specific inhibitors of HIF2 alpha, a protein I will draw to your attention again that we think is very important in causation of these kidney tumors in VHL patients, as well as other tumors in VHL patients. And these drugs are now available for evaluation in the clinic. And I'm going to show you some of our experience with two of these drugs. They're really very related drugs. And I'll show you a little bit about how these drugs are being developed in patients with VHL. As I said, these drugs were developed because investigators or scientists at a firm called Peloton collaborated with investigators at UT Southwestern to try and target this particular pathway and came up with a small molecule that very effectively and very specifically blocked this molecule called HIF2 alpha. Several candidate proteins or molecules were studied. And the first molecule that was chosen for further study in the clinic was something called PT2385. We at the NCI did one of the first studies with this agent, and certainly the first study in VHL patients with this agent. Anybody who had VHL had a measurable tumor in their kidney, meaning they had to have a tumor that was at least 1 centimeter and who didn't need immediate surgical resection of this tumor, they were eligible for the study. They should not have received prior treatment for their VHL or prior systemic therapy for their treatment for their VHL-associated tumors, and they should not have any evidence of metastatic disease. We put 4 patients on study, and in a fairly early notice that 2 patients with very high or the highest possible drug levels of this drug in their system had some tumor shrinkage in in the kidney tumors. And one of these patients also had shrinkage in their retinal angiomas. It's very hard to see these films, but I showed this just to let you know that we're very, very excited about what we saw in these patients. Those of us who are used to seeing these retinal angiomas in these kinds of pictures can spot the angioma. You can see it's right in the middle of that circle that I've drawn. These angiomas went away on therapy. And our ophthalmologists, Dr. Chu and Dr. Wiley, were just dumbstruck. They were so excited. They came down when they saw the first patient. They found me in my clinic and told me, we've never seen this before with any other drug. We're seeing this. So this generated a lot of excitement. But what we found was that this drug was not all we would like it to be. It seemed to have some effects, but based on our study, also another study that is being done in patients with sporadic kidney cancer, we found that not everybody was benefiting from this drug. And one of the problems seemed to be that if you gave the same drug at the same dose to 100 patients, not everybody handled the drug the same. Some patients had high levels of the drug, some people had low levels of the drug. And it was felt, and it was shown that many patients didn't have enough drug in their system to really be effective. Their body was just turning this drug over very quickly and getting rid of it. And in fact, this graph just shows you that if you had adequate levels of the drug in your system, and this is in the sporadic kidney cancer trial, then you had a much better, what we call progression-free survival, meaning you lived longer without progression of your tumor. That's the black line. And the red line shows that if you didn't have enough drug in your blood or in your system, then these patients progress fairly quickly. So with this in mind, the company Peloton and the scientists at UT Southwestern went back to the table and designed a drug that was a little different from PT2385, which they call PT2977. It's called MK6482 now, because the drug has been acquired by Merck. They found that this new molecule was much better handled by people. So if you gave it to 100 people, the likelihood that most of them would treat the drug similarly was much higher. So the drug level from person to person was much more even and much more comparable. It's also a very, very potent inhibitor of HIF2 alpha. And animal studies showed that this was a very effective drug in the animal models of kidney cancer that were studied. This drug was then taken into the clinic and looked at in patients with sporadic clear cell kidney cancer, where they found that a lot of the patients who had tumors, kidney tumors that were metastatic and had been treated with many other therapies in the past, showed tumor shrinkage. This graph you're seeing here is what we call a waterfall plot. The line in the middle is the baseline, and any of the purple bars that you see above represents growth of the tumor, and anything you see below, all the bars going down, represents shrinkage of the tumor. You can see that a lot of the patients had significant shrinkage of the tumors, and it looked like more patients had their tumor shrinking than didn't. Some of these responses were very durable as well. I won't go out to explain this graph in much detail. Suffice it to say that the longer the line, the more duration of treatment. So the patient has benefited from it and has stayed on treatment longer. And you can see some patients have stayed on it for over a year. The drug, more importantly, was very well tolerated. The most common side effect of the drug was anemia. And this was something that we would expect, because as part of its effect on HIF2 alpha, we expected that this drug would drop the levels of a protein called erythropoietin, or EPO, which is necessary to make blood cells. And we knew when that happened that the patients would develop anemia. In fact, when we see anemia develop, we know that the drug is, at least in part, doing what it's supposed to. Most of the patients with anemia are able to handle the situation fairly well. Sometimes they need us to replace this protein called erythropoietin, which is available for clinical use, and that usually takes care of the anemia. There were also a few patients who experienced low oxygen levels in their blood. The vast majority of these patients, it looked as if they had other respiratory or cardiac problems. And most of them, I think, were well managed. Some of them regressed spontaneously. Some of them had to have their drug stopped. But there were no other major side effects that were reported. Patients seemed to like this drug a lot more than they like drugs like pazopanib, vandetanib, and sunitinib. But again, this was in patients with sporadic kidney cancer. Doesn't mean that that will necessarily translate to patients with VHL. But we were encouraged enough by what we were seeing to then design a study in patients with VHL. This is a large global multi-center study. 11 centers took part in the study. We planned to enroll 50 patients. This was a study that started, I think, in 2018, mid-2018. Anybody who had diagnosis of VHL, had at least one measurable kidney tumor, and had no evidence of metastatic disease, and had received no prior systemic treatment was eligible for this study. And we're very, very excited about this study. The study has completed accrual. This is just to show you that the study opened initially at 11 centers, 8 in the US and 3 in Europe. In April of 2019, the accrual is complete. And soon thereafter, the drug, as I said, was acquired by Merck from Peloton, which originally made and developed this drug. Data analysis is ongoing. We'll hopefully have some data to show people in the not too distant future. But more importantly, what does this mean for patients with VHL? Can we get the FDA to approve the drug based on a study like this? Generally, if you want to take a drug and show the FDA that this is helping people, you need to approve it. You need a study that shows some kind of meaningful clinical endpoint. The primary data that we were looking for in the phase 2 study here is whether tumors were shrinking or not. And while we may be able to show that shrinking tumors are actually conferring a clinical benefit, that's not an ideal endpoint that the FDA likes to see. They like to see quality of life data. They like to see that patients are living longer or avoiding surgery or some such endpoint is usually required. Those kinds of studies generally, but not always, require a bigger study, a randomized study where some people receive the drug, some people don't receive the drug, so we can compare apples to apples. But there is a precedent for drugs being approved based on studies just like this, single-arm studies with no comparison arm or no control arm in the study. One of these examples is actually relevant to kidney cancer. Sunitinib, which is one of the drugs that used to be very widely used in patients with sporadic kidney cancer of the clear cell variety, was approved initially based on a single arm study. So all they showed was a lot of patients were having their tumor shrink. The FDA approved it, but said, you need to do a longer and bigger study to show that this is indeed benefit using some of the clinical endpoints that I mentioned earlier, but they did approve it. The second drug called avelumab, which is an antibody that targets key aspects of the immune system, was approved without the requirement necessarily for a larger study in a condition, rare skin condition called Merkel cell carcinoma. So this is possible, but there are specific circumstances that really lead to approval with a small study of the kind that I've shown. And this is generally a decision that's taken by the FDA. And they look at all the data, they look at whether or not, in their opinion, they can safely say that this drug is actually benefiting people and should be approved. So they have a long process, a very thorough process of vetting these drugs and vetting these studies to decide if and when to approve these drugs. So while theoretically possible, approvals of this nature are rare. One question that I've been asked many times that I thought I would address as part of my talk is, can VHL patients get this drug now? The drug is not FDA approved at this point for any indication. So they can only get access to this through a clinical trial. The clinical trial that I mentioned has been closed for now. It's possible that new clinical trials may be coming up, but there's no information that I have available that I can tell you, but I can't tell you based on the information I have whether a trial is forthcoming or not. As far as I know, there's nothing designed, nothing out there right now. If this drug is approved for another indication, let's say that there are studies that show that this drug is very promising and very effective in patients with sporadic clear cell kidney cancer, and the drug is approved by the FDA for that reason, it may be possible then to use the drug for VHL patients in the off-label setting. That is not the case today. Like I said, today, the drug has not been approved by the FDA for any indications, so it's not available to most VHL patients. I'm going to stop there and take any questions, but before I do, I want to thank all the patients and their families who over the years have supported research, both at the NCI and other centers, without which none of the development and none of the progress that we've made would have been possible. Also want to shout out a word for patients, care providers of these VHL patients, researchers, support groups, all of whom have really come together very well to really enable the progress that we've made over the last 2 to 3 decades. And this is an example of the kind of collaborators that I'm very, very lucky to work with. I haven't been able to include everybody that I've worked with, but I can't tell you how lucky I am to work with a wonderful, wonderful group of researchers and clinicians at the National Cancer Institute, without whom nothing that I've talked to you about so far would have been possible. I'm going to stop there and take questions.
Dr. Srinivasan, thank you. That was great. We have a few questions for you. How did you become interested in urologic oncology and specifically hereditary forms of kidney cancer?
Yeah. So I'm going to try to make it as brief as possible, but really it's going to be a little bit more involved than I think you may bargain for when you ask this question. So when I first took up oncology, I was actually very interested in bone marrow transplantation. And when I got to the NCI, I actually worked very closely with the bone marrow transplantation group. And as part of that group, there was very, very innovative research being done. And as part of my work with the group, we actually did bone marrow transplantation for patients with solid tumors. This was very, very novel at the time. This was in the early 2000s. Hitherto, bone marrow transplantation was reserved for conditions like leukemias and some lymphomas, as well as what we call bone marrow failure syndromes, where the patients didn't have functioning bone marrow or had problems with the bone marrow and they couldn't make the blood cells the way they should to survive. But then in the 2000s, late 1990s and 2000s, there was an effort to try and see if we could use bone marrow transplantation to benefit patients with metastatic tumors. And it looked like the population that was benefiting the most was patients with kidney cancer. And so that really got me seeing a lot of patients with kidney cancer. At that time, remember, there were very, very few treatment options for kidney cancer. Most of them didn't work in the vast majority of patients. So when I got into this kidney cancer field, what I saw was that there was very, very little happening for these patients. There was a lot of work going on, but very little had been translated into the clinic. And so to me, that was a challenge that I wanted to take up. And I joined this group in the urological oncology, Dr. Linehan's group, and was exposed then to patients with VHL, HLRCC, and other forms of hereditary kidney cancer. And I saw that, as difficult as it was to manage patients with sporadic forms of kidney cancer, even less work had been done on systemic treatment for VHL and other forms of hereditary kidney cancer. And really, that's what really piqued my interest, and that's what led me to do research in this area. And I can't tell you how gratifying it is to see the field develop, not just in terms of VHL, but also some of the other hereditary kidney cancer conditions that we see, as well as patients with sporadic kidney cancer that we see. A lot has happened in the last 15 to 20 years, and I like to believe that we've contributed to that progress. And it's extremely gratifying to see patients whom we used to tell, 15 years ago, I'm not sure there's much we can offer you to. Yeah, we have multiple options now, but this is how we think we should begin, and then we can go to X, Y, and Z. Sorry for the long-winded answer, but
no, that's great. And it actually goes perfectly into the next question: is what implications does your VHL research have on the general population, and why should people outside of the VHL community care about VHL research?
So if we did not study patients with VHL, if we were not doing research that was beneficial to VHL patients, we probably would never have understood how the more common forms of kidney cancer work. Today, we know that there are multiple forms of kidney cancer. Some of those, and the vast majority of those kidney cancers, are of what we call the clear cell variety. That's the variety that you see in patients with VHL. And it's only by studying patients with VHL that we had insights into these more common forms of kidney cancer. So I think what we learned from VHL has had a great impact on clear cell kidney cancer in general. But what it also did was help us understand that VHL is not the only kind of kidney cancer. There are other kidney cancers. They look different, they behave differently. By studying those now, we've really made inroads into understanding and treating other forms of kidney cancer as well. But going beyond kidney cancer, it's only because people have studied VHL over the last couple of decades, and at least, I wouldn't say it's only because, but at least VHL research has contributed to a significant amount of our understanding of how the body reacts to altered oxygen states. And that's got implications well beyond kidney cancer, other cancers, other processes, anemia, so on and so forth. So I think studying VHL has spawned interest in many directions, all of which I don't have the time to go over in this brief in a discussion session.
Has the coronavirus pandemic impacted the timeline for PT2977 at all?
I don't know globally what the effect has been. I can tell you at our center that we are continuing to see patients who are on study. Not everybody is able to make it because of the challenges with travel and other issues. But in those cases, we've done everything we can to support them. But I think it would be naive for us to believe that it hasn't had some impact. What the nature of that impact and extent of that impact is going to be, I think only time will tell.
We have one last question. Can kidney cysts become tumors? And if so, could PT2977 be used on those cysts before they become tumors?
Yeah. So in VHL, and remember this is only for VHL, pure cysts typically don't become tumors. We do see mixed cystic tumor lesions. So part of the lesion is a cyst, part of it is a solid tumor. Those can evolve to become almost entirely tumor. But today, in managing VHL patients, we don't directly target pure cysts. So if a surgeon goes in and sees pure cysts, he's not going to take those out. And similarly, at this point, I don't have data to suggest that by acting early and intervening on patients who have only cysts, we would prevent those cysts from becoming tumors. Now, it's entirely possible that if we see very promising data with PT2977 or some other drugs, then we could use them to prevent the onset of new tumors, regardless of whether people have cysts or not. So we could take patients who have VHL, we know they're going to develop tumors, or we know they've already developed tumors that have been surgically removed or managed otherwise. We can put them on study to prevent the development of new tumors. So an effective drug in the field does have the potential to be studied as a preventive agent.
Dr. Srinivasan, thank you again for taking the time to talk to us today, as well as for everything you do for your patients, especially the VHL community. We really appreciate it.
Thank you for having me today. It was indeed a pleasure and an honor. Thank you again.
And for everybody watching, if you are interested in learning more about VHLA's Surviving and Thriving Together series of events, please visit vhl.org/together. And until next time, stay safe. Dr. Srinivasan, thanks again.
Thank you again.